Good evening to all the NEET aspirants. This is your Prasanna ma'am from NEET World. So yes, in our last session from the chapter Anatomy of Flowering Plants, which is the last chapter we need to deal with from NCERT 11. Okay, what we have completed beta? Tissues, right? Tissues, both meristematic tissues and permanent tissues, both are done. What are the meristematic tissues that we have exercised? Can you tell me what are the meristematic tissues? Meristematic tissues based on origin, we have two types. Sit straight everyone, sit straight. We have two types of meristematic tissues based on origin. Primary in origin, secondary in origin. Primary meristems and secondary meristems. Primary meristems again based on the locations, we have two types. What are they? Apical meristems and intercalary meristems. Apical meristems, if they are at shoot or root, we have again two types, root apical meristem and shoot apical meristem. Secondary meristems or lateral meristems, based on the region they have present, we have two types of secondary meristems. What are they? Cork cambium and vascular cambium. Cork cambium and vascular cambium. And then we have what? And then we have learnt about different types of permanent tissues. We have two types of permanent tissues. What are they? Simple permanent tissues and complex permanent tissues. Simple permanent tissues are of what types? Parenchyma, collenchyma and sclerenchyma. Complex permanent tissues include xylem and phloem. If you have to consider mature or advanced division of plant kingdom, that is angiosperms, what are four types of tissues that are present in xylem? Tracheids, vessels, xylem fibers and xylem parenchyma. What are four types of tissues that are present in phloem? Phloem, sieve tubes, companion cells, phloem fibers and phloem parenchyma. This is what we have learnt in gist I have told you. Now we will look into tissue systems once. Tissue systems I will explain you in brief, and then we will look into the comparative analysis between dicot monocot root, dicot monocot stem, dicot monocot leaf. Okay? In that only I will explain you in detail what is the epidermal tissue system, ground tissue system and vascular tissue systems. So if you have to consider a mature plant, we have three types of tissue systems beta. Okay? Take down tissue systems. Tissue systems, we have three types of tissue systems. Just a second. Yeah, we have three types of tissue systems. Tissue systems include what? First is epidermal tissue system. Epidermal tissue system. Epidermal tissue system is usually single layered and it is made up of parenchymatous tissue. It is made up of parenchyma and it will be mostly single layered. Multi-layered you will see it in nerium and ficus. Okay? Multi-layered epidermis, multi-layered epidermis, you will see it in nerium and ficus. Okay? And what are those that you can observe in epidermal tissue system? Epidermal tissue system will consist of hair-like projections like root hairs, epidermal hairs, we will call it as epidermal appendages. Okay? And stomatal structures in leaf, okay? Trichome structures in stem. Sometimes epidermis is also covered by cuticle. Okay? Raghav, have some water. Okay? These are the constituents that are present in epidermal tissue systems. Clear, right? Next it is ground tissue system. Raghav, you can sit in last bench wherever it is comfortable to you. Ground tissue system. And third is vascular tissue system. Epidermal tissue system includes only epidermis. Okay? Vascular tissue system includes xylem and phloem. Xylem and phloem. What does ground tissue system include? Everything in the plant, all the components in the plant apart from epidermis and vascular tissues like xylem and phloem, apart from these two, all come under what? Ground tissue system. Okay, apart from these two, all comes under ground tissue system, all the components. That is the reason ground tissue system is also called as fundamental tissue system. Fundamental tissue system. So, fundamental tissue system includes what? First is, after epidermis, it is hypodermis. Hypodermis, general cortex, endodermis, which is the last layer in the cortical region. Okay, these are present. Okay, hypodermis, general cortex and endodermis. Fine. After this, in steel, okay, in steel, what are present in the stellar region? Pericycle, pith or medulla, and medullary rays, These are the components that are present in ground tissue system or fundamental tissue system. Okay. This is the gist that you have to know before you enter into the comparative analysis of dicot, monocot, root, stem, leaf. There I will give you the complete details of what will be the epidermal tissue system of the root, what will be the ground tissue system of root, what will be the vascular tissue system of root, stem and leaf. As of now, you have to know what is present in the epidermal tissue system, only epidermis, epidermal hairs, epidermal appendages, epidermal apertures. Okay. It is mostly single layered, in few plants it is multi-layered. Okay. And ground tissue system, if you see, ground tissue system includes everything except epidermis and vascular bundles. Vascular tissue system includes only xylem and phloem. Clear with this? Any doubt anywhere, ma? Clear, no? Next. Now we will see dicot, monocot root. Okay. In this you tell me under epidermal tissue system what comes? Only epidermis, right? Epidermis. And here epidermis doesn't have cuticle. Epidermis doesn't have cuticle in both of them. Now is the image clear, ma? Yeah. Epidermis doesn't have what? Cuticle. It is not uncovered by any cuticle. And it has unicellular hair-like projections called as root hairs. For both monocots and dicots, I am telling you. Both dicot and monocot, epidermis is single layered and it has unicellular hair-like projections. Hair-like projections called as what? Root hairs. And there is no cuticle. Epidermis is parenchymatous. Epidermis is parenchymatous. Clear, right? Okay, epidermis is parenchymatous. It has unicellular hair-like projections called as root hairs which facilitate absorption of water and minerals, and there is no cuticle in the epidermis. Clear, ma? Next. Next is the general cortex that is present. General cortex. Okay. General cortex. General cortex is also parenchymatous over here. General cortex is parenchymatous and it has intercellular spaces. Speaking about both dicot and monocot, okay, there are very little differences in dicot and monocot root, I will tell you. Till then you look at here, okay. So, cortex is made up of parenchymatous tissue, it has intercellular spaces, and last layer of the cortex region is what? Endodermis. Endodermis is the last layer of cortex region. Here you have to mark one peculiar point with respect to root. Endodermis will have suberant depositions in their radial and tangential walls. Okay, endodermis has what ma? Suberant depositions in their radial and tangential walls. Radial and tangential walls. Clear? What is the main activity of suberin? Suberin is a coating, okay. It will not allow the movement of water, okay. Till here from root hairs to endodermis, whatever water and minerals are absorbed, those pass in between the cells and through the cells, okay. In between the cells, within the intercellular spaces, whatever water movement is there, that is called as what type of movement ma? Apoplastic movement, okay. And the movement of water and minerals in the cell, okay, in the intracellular spaces is called as what? Symplastic movement. As here in the radial and tangential walls, suberin deposition is there in the endodermis. From endodermis, only symplastic movement is promoted and apoplastic movement is inhibited. Yes, yes, intracellular. Okay, clear? Any doubt till here ma? No, right? And all the tissue component is what? Parenchymatous only. Epidermis, cortex, general cortex, endodermis. It is parenchymatous tissue which is present, okay. And after this we have what? Pericycle, stellar region. After endodermis we have what? Stele. We call it as stele. Stele includes pericycle, vascular bundles, and pith, okay. Pericycle, vascular bundles and pith. So here pericycle is again parenchymatous. How vascular bundles are arranged? Vascular bundles are exarch in their arrangement. What is exarch arrangement? Okay, at the end, at the peripheral region, primary xylem will be there and maturation of xylem is towards the center, okay. Protoxylem, which is the primary xylem, primitive xylem, is towards the periphery. It gets matured to the center. Matured xylem or metaxylem will be at the center. So maturation is from periphery to center. Fine. Such a type of vascular bundle is called as what ma? Exarch arrangement of vascular bundles. And here there is one difference ma. One difference in between dicot and monocot. Here in dicot root, you can see diarch to tetrarch vascular bundles, means 2 to 4 vascular bundles will be there, okay. Vascular bundles here it is 2 to 4, diarch to tetrarch. Whereas in monocots, vascular bundles are polyarch, okay. And here pericycle which is there, in morphology we have learnt, in morphology taproot system if you remember, the lateral roots will originate endogenously from pericycle is the point we have learnt, okay. Lateral roots arise endogenously from pericycle. Here in dicot root, as cambium is present, okay, cambium is also originating from pericycle. But in monocots only lateral roots will arise from pericycle, cambium is not present, so cambium will not originate from pericycle. Understood or not? Okay. In monocots only lateral roots or secondary roots originate from pericycle. Here along with lateral roots what are originating ma? Cambium from pericycle along with lateral roots. Cambium also originates, vascular cambium. Clear? Pith is small and inconspicuous in dicot root. Pith is small in dicot root. Pith is large in monocot root. Okay, pith is large in monocot root. These are the three differences between dicot and monocot roots. What are those three differences, you tell me? What are the three differences? Yeah, vascular bundles. Number of vascular bundles in dicots is diarch to tetrarch, 2 to 4. Here it is polyarch, many. And second difference is, here it has small pith, sit straight first. Here in dicot root you have small pith, here large pith. Okay, and third difference is what here? Lateral roots and vascular bundle originate from pericycle in dicot root. Here only lateral roots arise from pericycle. Clear, right? These are the three differences, ma. Make a note of it. Rest apart from that, everything is clear, no? Epidermis, if you see, is there cuticle present in the root epidermis? No. What is the root hairs that are arising? Unicellular root hair projections. Second is general cortex is parenchymatous, it has intercellular spaces. Last layer of general cortex is what? Endodermis. In the radial and tangential walls of the endodermis, what depositions are present? Suberin depositions are present where water transport is inhibited. Okay, and then stele is there. Stellar region includes what, Nana? Pericycle, vascular bundles and pith. Okay, pericycle is parenchymatous, it is single layered. Vascular bundles are what mode? Exarch mode. Protoxylem is towards periphery. Metaxylem is towards center or pith. Okay, this mode of development from periphery to center is called as centripetal or centrifugal. Center. Centrifugal. Good. Fine, right? Any doubt till here? No? Completely analyze the root system, both dicot and monocot. Here you tell me what comes under epidermal tissue system? Only epidermis. What comes under vascular tissue system? Only vascular bundles. Apart from that, ground tissue system is there. Ground tissue system also called as fundamental tissue system. In root includes what, ma? Cortex, endodermis, pericycle, pith. This comes under ground tissue system or fundamental tissue system. Clear? Anything else to be dealt in the comparative analysis? I will give you 2 minutes of time, go through the images of your text. If you are having any doubt, just let me know. And here, the suberin depositions are called as what is? Casparian stripes. It is named after the scientist Caspary. Okay? These suberin depositions are called as Casparian stripes, named after the scientist Caspary. Here you clearly note down, ma. Clearly note down. Dicot root and monocot root. First is epidermal tissue system. Make a table as such. Okay. In epidermal tissue system of both dicot and monocot root, what is present, ma? Epidermis. Wherein what is absent? It is single layered or many layered? Single layered. Yeah. Single layered, and it is parenchymatous. And Unicellular hair-like projections, what are they? Are you writing or not, everyone? Unicellular hair-like projections, root hairs. Multicellular. This is the epidermal description, epidermal tissue system of root. Okay. Next is ground tissue system. Ground tissue system, also called as fundamental tissue system. Includes what, ma? First is general cortex, general cortex, parenchymatous, both in dicot and monocot it is parenchymatous with intercellular spaces. Clear? Next, after general cortex, what is present, beta? After general cortex, what is the last layer of the cortex? Endodermis. Here in the root, there is, in root anatomy, there is no hypodermis. After epidermis, directly it is general cortex, and after that you have what? Endodermis. Endodermis in both dicot and monocot, they will have what? Casparian stripes. Casparian stripes, which are made up of what? Suberin. In what walls? Radial and tangential walls. Clear? After endodermis, general cortex and endodermis, then what is included in ground tissue system? Third is from steel, pericycle is included, yes or no? From steel, pericycle. Pericycle, in both of them again it is parenchymatous. What is the primary difference, ma? Here from pericycle, both lateral roots and what is originating? Vascular cambium. Good. Here from pericycle only in monocots only lateral roots arise, because monocots doesn't have cambium. Clear? Fourth is what? Medulla. In fundamental tissue system in root, medulla or pith is small in dicots, large in monocots. Clear? Small in dicots, large in monocots. See, this stands as your standard reference if you write it in a proper way. If you remember this chart, it is enough. Those two diagrams which we have dealt previously and this chart, it is enough. Okay? Tell me. Diarch, tetrarch, polyarch. Diarch means 2 vascular bundles. Tetrarch means 4. Polyarch means more than 4 and 6. More than 6 you can assume. Yeah. Next you see it is what tissue system? Vascular tissue system. Yeah, vascular tissue system. Vascular tissue system includes only vascular bundles. Vascular bundles are arranged in what pattern ma? Exarch pattern. And they are radial in their arrangement. What is radial arrangement? No. Yeah, if you assume this as steel, okay, if you assume this as steel, xylem is followed by phloem. Xylem is followed by phloem. Again xylem followed by phloem. Okay. So here this is phloem. Phloem and xylem. Okay. Xylem and phloem are at two different radius. So here vascular bundle arrangement is what? Radial. Okay. Radial and open in dicots, radial and closed in monocots. What is meant by open? If there is a cambium that is present, if there is a cambium that is present in between xylem and phloem like this, which increases the girth of the vascular bundles, which increases the girth of the plant. Okay. If there is a vascular cambium which is present in between xylem and phloem, then that is called as what? Open type of vascular bundles. If there is no cambium between xylem and phloem, it is called as what? Closed type of vascular bundles. It is closed in monocots because vascular cambium is absent. It is open in dicots. Clear? Yes or no? Here, what is the type of vascular cambium and from where is it originating? In steel, what are present? I told you. Vascular bundles will be there, pericycle will be there, pith will be there. Vascular cambium is originating from pith or not? Okay. And remember beta, the tissue which is present in between xylem and phloem, this tissue is called as conjunctive tissue. Conjunctive tissue. So what happens is, from pericycle few of the cells will get meristematic and few of the cells in the conjunctive tissue will become meristematic and they start the cambial activity, secondary growth activity. Understood or not? Okay. So this happens only in the dicot roots, it is radial and open. In monocot roots it is radial and closed. Okay. And in both, what is the pattern of arrangement of vascular bundles? Exarch, where protoxylem will be towards periphery, metaxylem will be towards pith. Okay. One small diagram I will tell you, it will be easy for you. Protoxylem means primitive xylem is towards the periphery, metaxylem is towards the pith. Understood? In this way. Development is in this way. So it is centrifugal. Clear till here? Any problem, Raghav? Fine, right? So what are the primary differences? Vascular bundle is open here, closed over there. Vascular cambium is present, absent. Okay. It originates from pericycle. And here you can see diarch to tetrarch vascular bundles, here it is polyarch. These are three primary differences and these are the tissue systems. If you want to make a call to your parents, you can. Then? Clear, right? Here, dicot stem. Okay, focus here, else you sit in this row. Be here. Dicot stem. Okay. So here in the dicot stem, first you analyze the tissue system. When you have to compare root, stem, leaf, little complicated is dicot stem. Pay additional focus, attention and listen. Okay. So in the dicot stem, first is what? Epidermis as usual, right? Yes. Epidermis here is having a thick cuticle coated over it. Epidermis is covered by what? Cuticle. Cuticle has cutin. which is impervious for gaseous exchange. Okay, and you have unicellular hair-like projections in the root epidermis. Here you have multicellular hair-like projections, multicellular projections called as trichomes. Fine, right? Single layered. All the plants it is single layered except those two exceptions I told you. Here it is single layered. Okay, dicot stem, epidermis is single layered. It is covered by what ma? Cuticle, which has what? Cutin, which is impervious for gaseous exchange, and it has multicellular hair-like projections called as trichomes. These are called as epidermal appendages. Okay. Next to the epidermis, cortex is divided into hypodermis, general cortex, and endodermis. Okay, hypodermis, general cortex and endodermis. Abdul, any doubt beta? Clear, no? So here after epidermis you have cortex. Cortex has 3 layers: hypodermis, general cortex and endodermis. In root though you don't have a hypodermis, you just have general cortex and endodermis. So here hypodermis in dicot stem, it is made up of collenchyma. Remember, it is made up of collenchyma. What is the characteristic features of collenchyma? Collenchyma cell wall is made up of cellulose, hemicellulose and pectin. They can be present in the form of patches or arranged. There are no intercellular spaces. It provides strong mechanical support. Okay, they are present around the corners, and these are main compositions in the petioles of the leaf and stem regions. Okay, so collenchyma characteristics you have remembered, right? Here hypodermis is made up of collenchyma. General cortex is parenchymatous. Last layer of the cortex region is what? Endodermis. Last layer of the cortex region is endodermis. Yes, parenchymatous. But then endodermis here has starch sheath. Starch sheath. Okay, endodermis has starch sheath. Fine. After the cortex region, what do you have? Stele. Stellar region. Includes first pericycle, vascular bundles, and pith. Fine, pericycle, vascular bundles and pith. Okay. So here if you see pericycle pericycle is arranged in the form of semilunar patches and it is sclerenchymatous. Okay. Pericycle is sclerenchymatous. Sclerenchymatous. It has semilunar patches. Clear? Vascular bundles are arranged in endark manner here. The mode of development is endark. Okay, and here you can see conjoint, collateral, open type of vascular bundles in dicot stem. What does it mean? I will tell you. See. Meanwhile, you take down that. At the center it is pith, fine, and this is pericycle. Xylem is always towards the pith, phloem is towards the pericycle. Okay. Here if you can observe, xylem and phloem are at the same radius. Such type of xylem-phloem arrangement is called as conjoint. And there is a vascular cambium from the beginning. This cambium which is present in between xylem phloem called as what? Intravesicular cambium or vesicular vascular cambium. Intravesicular cambium or vesicular vascular cambium. Okay. And the cambium which is present in between two vascular bundles is intervesicular cambium. Intervesicular cambium. As such, this forms a cambial ring which permits stellar secondary growth. Stellar secondary growth. Clear, right? Understood the meaning of conjoint, collateral, open. So, in contrast to the mode of development in the dicot root and monocot root, you have observed exarch pattern, wherein protoxylem is towards the periphery, metaxylem is towards the center. In stem it is exactly opposite beta. Protoxylem is towards the center and metaxylem is towards the periphery. Okay? Protoxylem is towards the center. If I have to take this small piece, okay, it will be as such. Protoxylem is towards the center and metaxylem is towards the periphery, like this. Got it? So, here it is centripetal in its developmental pattern, where it is from the center to the periphery. Clear? Understood? Mr. Kunar, is it okay? Yeah. So, have you understood the meaning of endarch? In endarch, metaxylem will be towards, protoxylem will be towards, that is in stem. Okay. Conjoint, collateral and open. Clear, right? And pith is parenchymatous again here. Parenchymatous. Along with pith, in stem you also have medullary rays. Okay, you also have medullary rays which promote radial conduction, radial or lateral conduction of water, which promotes radial or lateral conduction of water. Once again, a quick gist from dicot stem. Epidermis over here is covered by what? Cuticle. It has cutin which is impervious for gaseous exchange, and it has multicellular hair-like projections called as trichomes. Okay, sit straight. Below the epidermis you have what? Cortex region. It is three-layered: hypodermis, general cortex and endodermis. Hypodermis is collenchymatous, general cortex is parenchymatous, endodermis has starch sheath. Okay, what is the last layer of the cortex region? Last layer of the cortex region is endodermis, remember. Okay, and after cortex you have what? Stellar region. Okay, stellar region includes pericycle, vascular bundles and pith. Okay, pericycle is sclerenchymatous. Okay, it is arranged in what? Semilunar patches, and vascular bundles are endarch, conjoint, collateral and open. In endarch type of vascular bundles, protoxylem is towards, metaxylem is towards periphery. Clear, right? And conjoint type of vascular bundles means xylem and phloem present in the same radii. Collateral, two vascular bundles are laterally arranged to each other. Okay, and open, vascular cambium is present. Here you can see cambial ring like this, whereas in root you can see wavy cambium, okay, which comes from pericycle and conjunctive tissue. And after pericycle it is vascular bundles, and next pith which is parenchymatous. Along with that you also have medullary rays which will promote radial and lateral conduction of water. Clear with the dicot stem? Any doubt anywhere ma? Now monocot stem will be very easy to you. Here itself I will tell you what are the tissues that comes under epidermal tissue system. Under epidermal tissue system in dicot stem, what are the regions that comes under? Epidermis. Only epidermis. Under ground or fundamental tissue system, what all comes under? Hypodermis, general cortex, endodermis and pericycle. I am talking about ground tissue system. That is the point. Many students will confuse between ground tissue system and stellar region. You people will think stellar region comes under vascular tissue system. No. Only vascular bundles comes under what? Vascular tissue system. Epidermis comes under epidermal tissue system. Apart from these two, everything comes under what? Ground tissue system. That is the reason it called as fundamental tissue system. You tell me now what are the tissue, what are the regions that comes under fundamental tissue system?
General cortex, endodermis, medullary base, pericycle.
Okay. Under vascular tissue system? Any doubt anywhere? Clear, right? Next, can we go to monocot stem? Yeah, monocot stem, listen here ma. Here also the epidermal tissue system or epidermis description is same as that of dicot stem. Okay. Epidermis is same as that of dicot stem. It has cuticle present and trichomes also. Below that you have hypodermis. So cortex here is just having hypodermis and general cortex. Hypodermis in dicot stem is made up of? Hypodermis in dicot stem is made up of? Hypodermis in monocot stem is made up of sclerenchyma. And general cortex is parenchymatous. General cortex is parenchymatous. Okay. So here you see beta. After cortex region there is nothing present. Okay. In monocot stem there is no endodermis, there is no pericycle, there is no medulla or medullary rays. Then what is present? How vascular bundles are present? Where is the vascular tissue system over here? Here vascular bundles originate in the general cortex itself, okay, and their mode of origin is schizolysigenous in origin. What does it mean? I will tell you. See, vascular bundles are schizolysigenous in origin and they are conjoint closed type of vascular bundles and then endark type of development. Schizolysigenous origin means in between the general cortex is what ma? Parenchymatous. So definitely there, listen first, listen, I will give you time to write. General cortex is what? Parenchymatous. So completely there is parenchymatous tissue only. Parenchymatous tissue will have intercellular spaces or not? Yes, it has intercellular spaces. So in between those spaces, and if at all few cells are dead, okay, in those spaces vascular bundles got originated, okay. Such a type of origin is called as what? Schizolysigenous in origin. And here how vascular bundles are present? Again conjoint type of vascular bundles, where xylem and phloem are at the same radius and they are closed, means there is no cambium in between xylem and phloem. Got it? And then endark pattern of development, that is metaxylem is towards the periphery, protoxylem is towards the center. Clear, right? Any doubt anywhere? Here you tell me, what are the regions that comes under epidermal tissue system?
Epidermis.
Only epidermis. What are the regions that comes under ground tissue system? Raghav? Hypodermis and general cortex. Apart from that, no other tissues are present. What are the tissues what are the regions that comes under vascular tissue systems, Abdul? Vascular tissue system in monocot stem. Vascular bundles, only vascular bundles. Clear, right? Okay. Once let us compare between dicot and monocot stem quickly. See, now if you learn this in comparative analysis only, it will be clear for you at the time of revision. Okay. If you go on reading this, it will not happen. Okay. For your exam, comparative analysis in anatomy is compulsory. Definitely there will be a question in comparison with dicot, monocot, root, stem and leaf. Okay. So pay utmost attention when we are comparing. Okay. First is what, ma? Epidermal tissue system. That's it. Here it is epidermis. It has what? Trichomes as epidermal appendages, and it is what? Parenchymatous. Yeah, cuticle. Uncovered by cuticle layer. When it comes to fundamental tissue system or ground tissue system, here you have what?
Hypodermis.
Yeah, hypodermis. Hypodermis, general cortex and endodermis. Pericycle, pith, yeah. Hypodermis is very good. Cool. Kunal, you can leave better. Endodermis has what sheath?
Starch sheath.
Pericycle has semi-lunar patches made up of what tissue? Sclerenchyma. And then pith is again parenchymatous. Medullary rays is also parenchymatous. Here if you see in monocot stem, only hypodermis is present. Hypodermis is sclerenchymatous, right? General cortex is, Mr. Raghav, general cortex is in monocot stem. Okay. Okay, and here there is no endodermis, pericycle, pith, medullary rays. Clear? Third is what ma? Vascular tissue system. Includes what? Xylem and phloem. How they are arranged? Conjoint, collateral, open, and endark mode of arrangement. Here it is conjoint and closed. How they are originated? Schizolysigenous in origin, right? And same pattern of endark arrangement. Clear? Dicot, monocot leaf and NEET previous year questions on this chapter we will look tomorrow in the next class. Till here, if you are having any doubts, you are open to ask. Both online and offline students, ma. You are open to ask any doubt. Dead cells are there, and wherever there are intercellular spaces, there is some gap in between them, no? Their vascular bundles will get originated. So clear, ma? Done with today's class? Once revise, any doubt you have, you can ask me, okay? So that's all for today. In our next class, we will look into the comparative analysis of dicot, monocot leaf and a gist of secondary growth. Secondary growth, anyhow it is eliminated for your NEET, but we will look it in periphery, okay, wherever connecting concepts are there, and then we will directly go on to the NEET previous year questions, okay.